Hello, this is Ms. Dagenford, and we are going to be looking at an introduction to cell signaling. And we're going to start with the simple ones and the basic concepts of cell signaling. So by the end of the video, you should have a good understanding of where this occurs, the basic ideas of how it occurs, and then some implications for what's happening. Like, why does this occur? What's going on? The nice thing is you'll start to realize that we're applying a lot of what we already know, which is nice. You should recognize a lot of components of this. So we're going to start with the simple ones. And one of them would be yeast. And yeast cells, they reproduce asexually, but they can undergo asexual reproduction, and that is when they exchange alleles with another yeast cell. But it's going to do that with a different type of yeast cell, meaning that they just have a slightly different version of the proteins. In fact, there's some idea that this is the kind of the beginnings of sexual reproduction. You know, we have a male versus a female, or a type A versus the type alpha. So we have type alpha, then we have A, and each of them is producing a protein that matches the other's factor. And so as the signals are given off and they bind, that's going to allow the two cells to come together, and then, yay, we have a new combination of alleles. Now, we already learned about the second one, bacterial quorum sensing. If there's enough inducers around, meaning that there are a lot of bacterial cells, then all of the bacteria are going to respond by creating new proteins. We saw that with luciferase, with pathogenic toxins. Those were not the nice ones. We even see this with the myxobacteria, these slime bacteria, and slime molds will do something similar, where if there's only one individual, they're not going to they're just going to go hunt and eat by themselves. But if you start sending out signals, they're going to come together and aggregate to create these interesting spore-forming structures, the fruiting bodies, and then they create these little and release the spores. So it's a way to kind of spread the spores farther distances, and also a form of reproduction. So this is similar to the yeast and the bacteria. They're releasing signals. Those signals are being received. So what about us? Well, our multicellular bodies are going to be a little bit more complicated. We're going to see these signals that can be local. They're going to send out signals, and they're going to go just to the cell next door, or a few cells over, or the cells that connect to other cells in the neurons. Or we're going to have long-distance signaling. We're going to spend some time focusing on that. Now, so let's take a look at the basic structure. Now, the basic structure here, and here's where you should start recognizing some components. So you should look at this diagram. Let's practice our, making sure we're familiar with everything. Here we would have our cell membrane with all of our little phospholipid molecules. Here's our protein, a signaling molecule, and you'll notice something that we've talked about before. Proteins have a specific shape. Now, when the signaling molecule binds to the protein, that's called reception. But here's the really complicated part. On the inside of the membrane, this protein is going to change, and as it changes, it sets off a chain reaction from molecule A to molecule B to molecule C, and it's just like you're sending a message. So up here, my little stick figure is knock, knock, knock on the door, but he can't get in, and that's going to be a real big key here. And then once that knock is heard, they're going to send the signals. Now, I put little hats on them to emphasize these are different molecules, and as they're different molecules, it's almost like the Pony Express, right? He's going to deliver the message to this guy's going to send the message to this guy, and then eventually you're going to get to the makers, right? The builders, who are getting that signal to either build something, break down something, convert something, transcribe something, translate something, do something. So we've got reception, transduction, you can think of this as converting the signal, and then the response. So let's look at some of the implications here. Only some cells make the receptor to the signaling molecule. So only some cells respond to the signal. That's really key to the whole endocrine system, the signaling pathways. If you don't have that receptor, then you're not going to get that message. In essence, you're just not going to hear it at all. Now, the shape of that protein is going to be the key, and we're going to look at a couple of vital ideas here in just a second. And then the messengers, this is going to be in multiple steps, usually. There are a couple of easy ones, but most of them involve multiple proteins and enzymes that are going to be sending the signal. And then the response, again, do something. Respond to this by actually changing something in the cell. So the signal, just as a big point, the signal is not going inside, at least with a lot of these. So let's look at a couple of really important key ideas here. Anything that changes the shape can affect the signal. So if it was originally like this, the receptor, and the molecule that binds to it fits in very nicely, if you have a change in the protein to make it look like that, then that might be enough that signaling molecule cannot do its job. And so then you get no response. And of course that would come about through a mutation in the protein. Anything affecting the strength of binding can affect the response. Now we need to dig a little bit deeper here, because how does this work? Well, again, it's a mutation, but this one's a little subtle. If there's a mutation, it may not affect the shape per se, but maybe it affects the binding of the signaling molecule to the receptor. So maybe we've got some more of these charges, the positive and negative charges, and so it binds stronger. And if that's the case, you might have a longer effect, you might have more of an effect, you might have less an effect. We can see this in some of the engineered forms of insulin that we can kind of play around with to see if it's going to last longer or stick around or have more of an effect. So anything that does that, that's kind of an important point. The shape, but also the binding strength can really play a role. So let's take a look at the receptors a little bit more in detail. The receptors are going to be in the membrane, but they're going to have an extracellular component and an intracellular component. So here we have a hydrophobic region, the nonpolar region inside. So what do these guys do? They bind to a specific signaling molecule. Now, I've been using signaling molecule. The fancy term is ligand, and that's going to be water soluble. Now, you can think of ligature, ligament. Remember, they kind of connect, and so ligand is going to bind to that receptor. So what happens to the receptor is it's going to change the shape on this inside portion. So let's do a quick model here. We have a receptor, and then the part that goes through the membrane. So this is outside, this is inside, and then we've got a part on the inside. When that ligand binds to it, the inside part is usually what's going to change shape in some form. And as that happens, it's going to then activate the responses. It activates the receptor. It'll then trigger this chain response. So one nice little tie-in, I hope you guys think about what's going to happen. These are proteins. We've talked about this before, but it's been a while. These are proteins, and as proteins, you have links of amino acids that then coil up to form that shape. So if we were to unravel this, we might get these long chains of amino acids. And then you would notice regions that are non-polar, that those would go inside. But let's think what that would mean. If we were to go back to the DNA, you'd have certain pieces of DNA that we know will lead to those nonpolar amino acids. So one thing that we can actually do is we can look at DNA strands and have a sense of where this protein is going to be. And a very classic signaling molecule receptor would be polar side that goes outside, a non-polar region that goes through the membrane, and then another polar region that might go on the inside. That tends to kind of be a standout. Oh, this goes through the membrane. Maybe it's not a receptor, but it's in the membrane. That's a kind of a nice pattern there. We can talk about that and unpack that a little bit more. Well, this is a good start. Let's start with the specific ones in the next video. Have a lovely day.